Hi, everybody. It's Keith Ligon here from the Society, and we are excited today to have our guest speaker, Kelly Gordon, joining us. We'll introduce shortly. Just before that, though, I wanted to get into an announcement, which is just to remind people we have our elections, which are now currently ongoing and open. The last day that you can vote is 2 days away. So Friday at 11:59, Aaron will be closing the election. And so we have really excellent panel of candidates for everybody to select from. And so really want to encourage people to reach out. And thanks to everyone who did join the election and is running. We really appreciate that as well. So just again reminding Friday night, get your votes in to make your voice heard, and we'll be following up after that with announcements of the winners. So today we're really lucky to have Kelly Gordon joining us and sharing on her experience out in the field. We were talking about of regulatory and helping people with functional precision medicine testing as well as other testing navigate regulatory processes, trying to get approval and get precision medicine in the hands of patients. Just a bit of background. She's the founder of Boudicca DX, which is a global precision medicine company where she supports these types of filings and regulatory strategies. So really great at thinking out those strategies, which she'll share with us today. Just her background, she has a background in pharmacology from Duke and University of Arizona, where she had done her education. She's passed through TGen, in Arizona and done many types of CAP certified and CLIA accredited laboratory sort of filings and LDT launches, where she was a molecular technologist as well, directly involved in those, and has done companion diagnostic IVDs for oncology treatments at Roche Diagnostics and other pharma companies. And she's now been in regulatory affairs for 10 years and holds a regulatory affairs certification in devices from the Professional Society for Regulatory Affairs. So really a great resource. We're really pleased to have you, Kelly, speaking with our membership. And with that, I will let you go ahead and share your insights. Thanks so much.
Great. Thank you so much for the invite. Go. Can everyone see okay? Perfect. That's good. Good start. So today I'm going to talk about how functional precision medicine testing must navigate both drug and device regulations. This is what makes what you do kind of complicated because you're dealing with both drugs and devices, and we'll go into that. I'll first talk about the US FDA investigational clinical trial assay regulations. Then I'll dive into Europe. I started off very much US centric. And lately, a lot of trials are going on in Europe. I'm not focusing on the rest of the world also, but we're seeing, more trials in China and more trials in Australia. So it's important to kind of be cognizant of those international regulations too as you start developing these tests. I'll end with some considerations for commercial. The bulk of this is going to be how you navigate clinical trials, but I think it's important just to think with the end in mind if you were going to take some of these products through FDA and actually market them, put them on the market, and they would be regulated, what that would entail. So, as you all know more than me, functional precision oncology is when you're taking these patient tumor cells and you're treating them with therapies. And the idea is really to provide immediate, translatable, personalized information to guide therapy. So these are for therapeutic decision making, and that's why they become under regulations. Precision oncology. So a lot of what I worked on has been static features of tumors. So companion diagnostics, a lot of the time, for example, focus on genomic alterations like NTRK fusions, ROS1 fusions, EGFR mutations. So the idea of this functional precision medicine, which I'm really excited about with what all of you are doing, is really giving more information to patients and more potential options. The goal here is really to match cancer patients to the therapies that are best for them using functional features. And those therapies could be approved therapies that are out there already that are regulated by the authorities or investigational therapies. And with that comes a lot of regulatory burden. So if you're dealing with, authorized, approved therapies, you have to go into FDA's website. So here in this slide on the left, for example, you can look at the, indications for FDA approved drugs, the indications for European Medicine Agency, so EMA, authorized medications. And then a lot of times we're dealing with investigational therapy. So these may be therapies that are under development. So for example, if a pharma partner, a pharma sponsor comes to you and wants to use your functional precision medicine test, they may have a therapy that's novel and it hasn't gone through FDA regulations. So these are important considerations when you're developing the clinical trials, the therapies that you're looking at for these tests. Are they approved? Are they investigational? So that's one component on the therapeutic side of the fence. But then the device. So the other complications. So I really like this example from this Nature paper where it shows really the complexities of a device. And this is also regulated by FDA. So when you use these functional precision medicine tests, every component is regulated by FDA when you put it into a clinical trial, for example. So you can see here, the enrollment of those patients, what are the inclusion criteria, what's the informed consent look like, what samples are you taking from them, for example, tumor samples, blood samples. How are you using those samples? So that's all regulated by FDA, for example. Then all the controls around it. So how much sample do you need? What's your testing failure? What's your format? Are you using a genomics panel, for example? Are you using flow cytometry? All the methodology also comes under the purview of FDA. There may be tumor boards. So there's individuals that may be involved in making those decisions based on that biomarker data that you're producing with these devices. That's all regulated. And then all of that comes together to present information to make a therapeutic decision. And that's when it becomes interventional and is subject to regulations. So you can see some of these assays can be quite complicated from a device perspective. And there's a lot of things to consider: the operators, the format, the software, cybersecurity. And that's where it can become complicated when you start to take these to the regulators. So in terms of the regulators, you may not be familiar with who they are. So in the EU and US, we're usually dealing with CBER if we have a biologics, or we're dealing with CDER if we have a drug. A lot of times, most of my projects, especially in companion diagnostics, are with CDER. And CBER, we have some companion diagnostics as well, but most of the time I'm dealing with CDER on the drug side. In Europe, it's EMA, so European Medicines Agency. And they're very different from FDA. So there's less targeted therapies, for example, in Europe. There's other considerations when you're doing trials. Also, at the country level, we have national competent authorities in Europe. So there's an extra level of regulation when you do clinical trials. And so, for example, if you're doing a clinical trial in Europe, you're more often dealing with that national competent authority. And when you've collected all that data, that may be when you go to EMA. So one of the important things is, these are drug regulators. They may not be familiar with devices. So a lot of what I do as a consultant is help explain the device in the drug submissions, because you have regulators that may not understand flow cytometry. They may not understand next generation sequencing. So doing a good job of informing the drug side of what the device really looks like, making it easy for them. And then on the device side, because you also have to consider that, it's actually a bit simpler. So in the US, we have one center, so Center for Devices and Radiological Health. So they regulate all the devices in our country. Unlike having CDER and CBER, where they have differential regulation, we have one center that regulates devices. It gets a little bit more complicated in the EU, which I'll go into later, where there's notified bodies that actually take tests through marketing. And there's some considerations for that. When you're dealing with the device authorities, it's really important to inform on the drug. So they might be experts in flow cytometry, next generation sequencing, and PCR, but they may not understand the drug development. They may not understand, why are you using this test to select therapies? What therapies are you looking at? Where are you with your clinical development? So one of the big pieces advice is always remember your target audience could use sometimes a little bit more education. So spending the time describing the device very simply. So, think of someone that doesn't know your device that you've made and your test that you've made, and really breaks down the components so that those drug regulators don't get hung up. And you'll see what a hung up means in terms of a clinical hold if they do have questions in a minute. So I really believe for these functional precision oncology tests to improve patient outcomes and become a standard tool, you have to learn how to navigate the regulations. If you can't get trials up and running, if you can't navigate drug and device regulations, you can have clinical holds. And then unfortunately, these tests sometimes will not get to patients. So the idea of today is just to give you some insights. I'm happy to take questions at the end. I'm happy for anyone to email me if there's any questions that are lingering. And the idea is just to give you some tools that maybe can help you move your programs faster if you are dealing with device and drug regulators. So I like this slide because I feel like in this world of regulatory, we use a lot of acronyms, and it can be really confusing when you're trying to figure out what are the regulatory requirements for the testing that you're doing. And this is really sort of the hierarchy that we see in the United States. So we have research use only tests, for example, at the bottom, which are exploratory use most often for biomarker evaluations. Sometimes I like to say when we're playing around with our tests to figure out where they might be most useful and we're collecting research. That's what you kind of think about in terms of research use only. And then as you move up, when you start to make patient management decisions with tests, that's where CLIA kicks in. So in the United States, if you're using a test to make a decision with a patient, it's typically, it has to be done in a CLIA laboratory. So they're regulated under CLIA. So if you're using a functional precision medicine test to make a decision, you do have to think about the CLIA laboratory. Then we have other accreditations like CAP. We have New York State CLEP. So if you're dealing with New York State patients, you have to think about the requirements from New York State CLEP. So these are additional things that we think about when we're using these types of tests. Then once you've thought about all of that, then you have to think about FDA. So FDA regulations can kick in, which I'm going to go into in more detail. Investigational use only regulations. So that's when you're using tests in clinical trials. Are they subject to these regulations, which I'll go into in more detail? And at the top of this, where you see most companion diagnostics, are tests that have gone through full FDA clearance, FDA approval. They've looked under the hood, they've looked at all the software, they've looked at all the reagents, they've looked at all the instruments, and they've blessed them. And that's when you get clearance and approval and can use these tests in the market. And that's really the highest level of regulation that we have in the United States, is going through FDA clearance or approval. Where I see most functional precision medicine testing are in these buckets. So for research use only, so for exploratory clinical research, for example, which generally is exempt from the investigational regulations. They often are for clinical research, going to IRBs, not necessarily going through FDA. And a lot of tests will be, RUO, and there's not many requirements, often just GCP considerations when using clinical trial samples, for example, for clinical research. I see functional precision medicine tests commercially used, so in CLIA certified laboratories. And I'm going to go into this in a little bit more detail. FDA does have enforcement discretion for CLIA tests. So they're not regulated directly by FDA on a continual basis, but they send things like warning letters. So it's always really important to just be cognizant, have backup plans, for example, if you're using research use only reagents in LDTs, because if FDA, for example, shut down a manufacturer because of an RUO reagent, that could affect CLIA labs. So that's an important consideration. I won't go into too much detail on that today. Investigational use only, I'm going to go into a little bit more detail on the next few slides. And then in Europe, we have CIVD tests. So these are the types of functional precision medicine tests I've come across in terms of the different regulatory status of these tests. So as you probably are all aware, FDA attempted to regulate CLIA LDTs. So in May 2024, feels like it was so long ago, they finalized the LDT rule, and they just added these words, including when the manufacturer of these products is a laboratory. And then everyone in the CLIA world got very grumpy because FDA was going to regulate CLIA LDTs, and effectively they would all become cleared and approved IVDs. And as you can imagine, this would have been extremely burdensome. It can be expensive to take your test through FDA to generate all the data, which I'm going to go into. So a lot of laboratories pushed back, and there was a lawsuit. So the American Clinical Laboratory Association and the Association for Molecular Pathology, they sued and they won. The rule was vacated on March 31st, 2025 in a Texas court, and the final rule was no more. So it's officially been rescinded, and now we are back to enforcement discretion. But one thing I can say is after this, change to the regulation or throw out, I have seen a little bit more scrutiny when I'm taking CLIA tests to FDA, and I'm going to go into that in a little bit more detail. As you can imagine, there was a lot of resources at FDA spent on regulating LDTs. This was thrown out pretty quickly, and one could imagine you may have some grumpy members at FDA that still feel like LDTs and those components, research use only, reagents, instruments, should be more regulated. And in fact, on March 31st, the same day that the judgment was thrown out, there was a warning letter that was sent to a manufacturer on the exact same day, saying that they, were misbranding the advertising of their research use only reagents. So even though we're not, formally regulated by FDA, it's still really important to look at tests in CLIA laboratories and just make sure you have backup options. So one thing also that was sort of a misconception after the CLIA LDT rule was thrown out is I work with a lot of pharma on, precision medicine tests, patient selection tests, companion diagnostics. There was sort of this misconception in the field that now we didn't have to worry about CLIA LDTs in our drug trials because the final rule was gone. Well, that's wrong because when you submit an IND to FDA and you say you're using a CLIA certified laboratory for your testing, you are subject to FDA regulation because you're putting the test into an IND application to the agency. So just because the rule was thrown out by by the courts, that didn't mean that these tests in CLIA labs wouldn't be subject to FDA regulation if you put them into your INDs. And shortly after all of this, there was another initiative at FDA, ELSA. So I always say I got froze by ELSA now when I have an AI response from FDA. So they use ELSA, this generative artificial intelligence tool that was released in May for the reviews. So when you send a protocol, when you send an IND to FDA, they use AI to review it. And so shortly after this, one of my clients was using a testing in a CLIA laboratory, and they received this output, which you've seen I've boilerplated and looks very much like a script that came from an AI generation. So in this protocol that was submitted to the IND, there was inclusion-exclusion criteria that, had a biomarker criterion. And so FDA said, give this request for information back saying, please discuss the rationale of the output of your assay. Please provide data why you're using this assay. And then the key one for all of us today is please discuss the regulatory status. So at the very bottom, it says, is your assay cleared or approved by CDRH with an active IDE? To which my client said, nope, we're just using a CLIA test. We don't have an investigational assay. And I'm going to go into that in more detail. So you can see that with AI, with changing regulations, we still really need to think about, the assays that we use in our protocols, because it's even easier now for the AI to run through all of those documentation that you send to FDA and, push out responses like this. And this is an example that I've seen numerous times on submissions since the deployment of AI. So what does this mean? Why do we care about investigational device exemption regulations? So this is where it does get very confusing to people working on tests that are used for patient selection, patient decisions, drug development, because they are subject to what we call these investigational device exemption regulations. And if we're exempt, and I mentioned earlier the example of research use only, then we don't have to follow these regulations. But often, especially for functional precision medicine testing being used in protocols and INDs, for example, it isn't, you can see this fourth criteria, not used as a diagnostic procedure without confirmation by another medically established diagnostic product or procedure. So if you're using a test to make the primary decision in a trial, for example, that's when it triggers. It's either going to be probably a non-significant risk or a significant risk. And so a non-significant risk, that is when you have a test, for example, being used for patient selection that poses a minimal risk. And I'll go into more details of how we determine that. And this is what you want. You want it to be a minimal risk because there's no investigational device exemption application, which I'm going to go into more detail of sort of the burden of that. And really all it is appropriate labeling, appropriate monitoring, appropriate IRB approval. So a non-significant risk is very low regulatory burden. There's just some extra responsibilities, and it takes, this test potentially in a CLIA lab to become a non-significant risk investigational assay. Now, what you often don't want to get is an investigational device exemption requirement. So FDA can come back and say, we consider your assay significant risk. I see this, for example, with flow cytometry assays, with minimal residual disease assays, and AAV gene therapy assays. We still often get investigational device exemption requirements. And why do we get those? So not only does FDA look at the risk of the assay and what a false positive and false negative mean in the context of the trial, they're also curious about technology. So if there's any reason that they want to understand more of the technology or have more control around that, that's when you can be asked for an investigational device exemption application, where you actually have to submit all of your validation data, which I'll go into in more detail, to the agency, and they must approve it. And why this is a concern is this can delay you, because if you don't get approval of your investigational device exemption application, you can't test patients with your functional precision medicine test, for example. So it's really important to know whether you have a potential investigational device exemption application requirement. So how do I do that? How do I know if it's potentially a significant risk and is going to require an IDE? Because you don't want to get a study may proceed letter when you've submitted an IND that says an investigational device exemption application is required, because we know we have to submit that. There's preparation time, there's FDA review time. So how do you go about actually determining that? Well, what's nice is we have a streamlined pathway, and a lot of sponsors forget about this. So this guidance on the left, if you Google this, this will come up. It's a streamlined submission process for study risk determinations. And I'm going to go in what the content is in a minute. But this is a way that in your IND, you can proactively lay out the risk of the device and propose it's a non-significant risk. And it's nice because it's evaluated in parallel with the IND. So you'll know immediately whether it's considered a significant risk. I always recommend this because CDER and CBER, for example, on the drug side of FDA, will have a consultation with CDRH very early during that 30 days, so it makes everything faster. If you filed an IND and you don't have the opportunity to do this, or you forgot, there's clients that come to me because they forgot and they got the letter, then you can go to CDRH directly. You can submit a submission on the right. We have the guidance that you would follow, and it's the exact same content, which I'm going to go into, but you're submitting it to the device side of FDA. I try not to use this because it takes up to 90 days often. And with FDA shutdowns, sometimes these go to the bottom of the pile. So you could be waiting 90 days for a decision to even know if you need an IDE. Then you have to make the IDE in another 30 days. So you could be looking at months added on. So I highly recommend, if you are filing an IND, if you're working with pharma partners, that you integrate the device into that filing to make everything faster. So what is the study risk determination? Well, it's really looking at the testing itself. So a device description, what type of test it is, as well as the risks in the context of that study. So these are the 4 questions that we answer from the guidance. So the first question is, will the results from that test lead to some study subjects foregoing or delaying a treatment that's known to be effective? So does a false positive mean that a patient wouldn't get, for example, a standard of care treatment? Then that could be a risk associated specifically to the device. The second one is, will use of the results from that test expose study subjects to safety risks that exceed the risks encountered with a control arm or a non-trial standard of care? So this is all about the safety. Will a false positive mean that the patient is exposed to greater, potential for adverse events as compared to standard of care? A lot of projects I worked on, there may not be standard of care. They may have exhausted all treatment options at that point. So you would fold that in. You would say it's a relapsed refractory population, for example. So the consequences of a false positive are that they would be going into an investigational therapy trial, for example, versus, not receiving a treatment. So you want to use your protocol to really answer these questions to show why the assay itself is not posing any additional risk. The third one's all about the test itself and why you're using it. So if you're using a functional precision medicine test, why are you using it? What's the algorithm? What's the outputs? What's the reason for the test being in the clinical trial? So that's more of a mechanism of action biomarker piece. And then the fourth one's all about sampling. So If you're using tissue, if you're asking, for example, for a fresh tissue biopsy or a fresh bone marrow, FDA could consider that to be a significant risk if that is only being used for the investigational testing. And so that would be a potential risk where they may want an investigational device exemption. There are ways to minimize that potential. Like, for example, if you're using it for other PD assessments and it's not just being used for investigational testing. So it's not always the case that if you answer any of these yes, that it's an automatic significant risk. It's just you need to provide the responses, give them to FDA, and provide your rationale why you think it's a non-significant risk. So I always recommend doing this with FDA through the IND because in my experience working on CDX companion diagnostics, for example, IRBs sometimes can be more conservative. And so I found it easier to go to FDA to get the study risk determination notification and put it in the IRB submissions versus adding extra time. You could go to a central IRB and they say, we think this is a significant risk. You need to go to FDA, and now you've added extra time. So the idea of this is put it in the IND, do it quickly. If it's a significant risk, it's a significant risk, right? There's nothing you can do. At least you know early so that you can plan and reduce some of those timelines. It's better than not knowing it's a significant risk. These are the two answers that we get. This is the boilerplate language. So on the left, this is what we want. We want them to tell us we don't consider it significant risk. It's a non-significant risk device. On the right is where they do consider it a significant risk and they require an IDE. So at least you get an answer early on what's required for the assay. And now I'm going to go into sort of what an IDE looks like. A lot of people are scared about investigational device exemptions, but it's more if you do a good job on the investigational device exemption and you're clear with your assay, they're easier to get through FDA. It is challenging in some cases, but I think a lot of it is just clearly describing the assay and the validation that's been done for the assay. So in these applications to CRH, so again, if you've got significant risk, if you are required to submit this application, the best way to think about this is it's like an IND for an assay. So when you do an IND for drug, IDE is analogous for the test. So because of that, we want to give FDA as much details as possible. So a list of the reagents, a list of the instruments. If they're serial controlled, add that, right? That's a level of control. So everything you can provide about the device, sample to result, just like in the figure earlier, which I think is a really good figure showing all the steps. I always recommend, having a figure with every component in it. And then report prior investigation. So all your analytical validation data that you've done, your clinical investigational plan, your labeling, your informed consent form, and then there's going to be a 30-day review. This is a highlights of what goes into an IDE, but you can go to this checklist and these slides will obviously be provided after, and you can see the long list. And I usually tell people just download this list. It can be your traceability matrix to make sure that you have everything that goes into the IDE and include it in your submission because then you show FDA, look, we tried to follow your checklist. We think we have everything in there. The other big thing that I see a lot of issues with the IDEs getting through is just not using the appropriate FDA recognized analytical validation terminology. So it's really important. Is it a qualitative assay? So for example, if it's a patient selection assay, enroll in trial, don't enroll in trial, it's probably qualitative. So you want to do all your validation in that way because it's going to be less burden. And I won't go into too much detail on the different ways to validate today, but just to give you an idea to keep this in mind. Semi-quantitative is more like you have ordinal categories like low, medium, high. So there's 3 buckets, for example, and then quantitative is you're actually, reporting specific metrics that you're using, like certain levels of biomarkers. There's a lot more burden to report quantitatively than qualitatively. So for example, if you're doing a precision study, if it's a qualitative assay, FDA is going to be looking for positive % agreement, negative % agreement across those 2 categories versus quantitative, they may ask for some more. And then in terms of the basic requirements, they want to see precision. So repeatability is, I always say, if you haven't done repeatability, just don't even go to FDA because they want to see your assay under identical conditions. And I mean identical. So anything you change, they're like, where's the repeatability? I'm not reading anything else. So you want to make sure you do that. And then intermediate precision, you can't do everything. You have small budgets. So I really believe in fit for purpose. Go through your assay. What do you think are the critical reagents, the critical instruments? Rationalize that. Say, we decided to look at these reagents because we consider them critical and include that in the IDE. This isn't the level of what you need for a marketing submission with FDA, but you just need to give them enough and enough rationale that this will support the use of your assay in the clinical trial. Analytical accuracy, it's not necessarily required if you have a novel test. Obviously, if you have a novel test, there's no reference method for you to check yourself. So if that's the case, you just provide that rationale to FDA. And then any other additional data you have, they want to know what you consider a limit of detection for your assays because they want to make sure that, samples close to those limit of detections, for example, are included in precision studies to see how they flip. And then the cutoff. So if you have developed this precision medicine test with a certain cutoff based on training sets, for example, in certain biomarker levels, provide the rationale. So the idea is to give them enough so they don't hold you up with requests for information or don't understand like what analytical validation you're doing and what the rationale was. I think this is honestly the best way to sort of mitigate the risks of these IDEs is really just using the language that FDA expects and really thinking about rationalize. You do stats are good, right? If you have statistical rationale, you should obviously include that. The more is better when you submit these IDEs for your tests. The other thing that you can do is you can actually do a free submission to FDA called a pre-sub. So if you're not sure what should go in an IDE and you have time, a lot of my projects we don't have time, but if you have time, I highly recommend this, where you know you have an investigational device exemption, you schedule a pre-sub with FDA, and you propose the studies, the repeatability, the intermediate precision, the device description. They'll give you feedback so that it can mitigate your IDE. And you include this feedback in the IDE application. What's really nice is there is a pre-sub template now. It's an interactive PDF that you see on the right. And there's a link here. So it's really easy to pop in your analytical validation plans and ask FDA for feedback. Usually you get feedback within 70 days or within 5 days prior to a scheduled meeting. Even with all of the sort of revolving doors at FDA, they're still sticking to these timelines. I haven't seen any budge. So if you did want to, go talk to FDA before an IDE, this is a good opportunity. That is free. So I think take advantage if you can. The other thing is, if you're dealing with a very novel assay, we do have this breakthrough device designation pathway for devices. I have used this for companion diagnostics. It speeds things up. So it allows you, for example, to have faster submissions. So if you have really good clinical feasibility data, technical feasibility for your functional precision medicine test, if you're thinking about taking it through IVD marketing, which we'll talk about later, this might be a good thing for you to get to have interactive discussions with FDA. As I started the talk, you're developing these novel tests. And so there is going to be some negotiations needed with FDA because it's novel, right? And they don't have the answers for how to develop these either. So taking the opportunities of having a pre-sub, doing a breakthrough device designation allows you to at least start the education process and the interaction with FDA. So this is also free if you get it. Within 60 days, you submit all of your feasibility data, your device description. And if you meet these criteria, which are on the right, FDA could grant you this and it allows you to have more interactive discussions with FDA. So I would recommend looking into this if you are, in a situation where you have the data, you do qualify and you want to have that relationship with FDA to have more interaction. So that was all FDA, which was a lot already. So if you make it through FDA and you are thinking about Europe, and again, I am seeing a lot of people go into Europe. There's less, approved therapies there. There's lots of clinical trials. There's lots of research. So I think it's also important to understand EU regulations. So in the EU, they don't use the same terminology, of course. So it's not investigational, it's not non-significant risk, there's no IDEs. They have their own terminology. So one of the first things to do, and hopefully you'll learn some here today, is what are the words they're using so that when you go to the EU, you're using those words. So you don't get requests for information and issues because we're using different words. So anything that's used interventionally in the EU in clinical trials to influence patient care decisions used to guide treatment is considered interventional in the EU. They don't have CLIA This is a good way of thinking. It's similar to US CLIA, where if you're making a patient management decision, you need a CLIA lab. So in the EU, if you're making a decision on a test, it's considered subject to EU IVDR, which is their regulation. What's interesting in Europe is they actually consider venipuncture to be invasive. And I always bring this up because it shows you the differences between the two geographies. In the US, venipuncture is not considered invasive. So the question 4 that we talked about for study risk determination, that's not considered an invasive sample type. So we don't have to worry about that posing a risk. But in Europe, it is. There are some initiatives to change this. So functional precision medicine testing, because of what we do, we're making treatment decisions. It is likely to be interventional and subject to EU regulations if you're using your tests on EU patients. So that's something to consider, if you're involved in clinical trials, even if the samples are sent, by the way, to the United States, you're still subject to these EU IVDR regulations. So one of the important things in Europe is to really understand who you're dealing with. So we have EMA, which I talked about at the start. We have the national competent authorities at the country level, but then these notified bodies. Well, it's really interesting in Europe. The notified bodies that actually regulate the tests and take it through CE marking, they're for profit. So they sign, agreements with manufacturers and they take them through the process of CE marking. That's very different than FDA. Obviously, CDRH, who we talked about before, is not for profit. So why do I bring this up? Well, there's a disconnect. So CDER and CDRH, they'll talk to each other. As I mentioned, during INDs, they'll have consultations. The notified bodies are not talking, for example, to EMA and national competent authorities during this entire process. So you have a disconnect in terms of the device and the drug development, for example. So it's really important in Europe to really be clear and detailed about the tests in the drug submissions, because there often are not device experts included because of the nature of how it's set up in Europe. When we submit for a clinical trial authorization in Europe, there's a cover letter where you check the test that you're using, for example. So this is what is checked when you submit a clinical trial authorization in Europe. So I'll go into detail on what these terms mean on the next slide. So you do have options in Europe for EU IVDR. So the way to think of a CE marked IVD is similar to FDA. So I mentioned, class 1, class 2 earlier. We were talking about ID exempt as a bullet point. That's marketed products in the US. They're called CE marked in Europe. Most of your tests that you're working with are not going to be CE marked. You haven't gone through the entire process. So that's why that one doesn't have a star, because it's usually not an option. This would be, for example, if you were using tumor profiling tests or that were CE marked, you wouldn't have to comply with these additional regulations. So the best way to think of a performance evaluation application, it's very similar to an investigational device exemption application in the US. It's a lot of documentation that is submitted to the national competent authorities, and I'll give you a list of that type of documentation next. And then the other two options, which only can be used if they're in Europe, are in-house IVDs in local laboratories. So if it's a hospital doing functional precision medicine testing in Europe, they could follow these regulations of being compliant with Article 5.5. It's similar to CLIA in terms of the requirements that they follow and that in-house approach. And then in certain countries like Germany, Netherlands, you can also be considered an in-house IVD provider if you're a CRO. So there are options if your laboratories were in Europe, if your companies are in Europe, you do have in-house and then or performance evaluation applications. But if you are testing samples in America from EU patients and making management decisions, that's where these performance evaluation applications can kick in. And they're very long. So unlike FDA, which has 30-day review periods, Europe can have up to 9 months. And it can slow a lot of pharma sponsors, for example, down. So it's important to know if you're working with EU clinical trial samples, what the requirements are. Again, if you want to use in-house, the lab must be in the EU. If you're doing testing in the US, you cannot leverage the in-house pathway. In the US, obviously, we have the performance evaluation applications, as I mentioned. If you do have a laboratory in the EU, and I do have clients that are building laboratories in the EU to circumvent this. So, for example, they were doing testing in the US, but now they are building labs in the EU because they want to be faster because they can comply with Article 5.5, the general safety and performance requirements, as they call them, and set up testing in Europe faster. So this regulation is really impacting how people are even planning their business operations. What's required for this in-house again is general safety and performance requirements, so risk management system, design, manufacturer, performance requirements, and all the information obviously has to be in file. So it's very similar of thinking of a CLIA lab where everything's on file. There could be inspections by the authorities, so they want to have all the documentation on file. One way to quickly meet the quality management systems is to have ISO 15189. It is a recognized standard in the EU. So for example, those clients that are building laboratories in the EU, they're trying to comply with ISO 15189. The regulation says or, but they ask you for it. So if you go to the authorities and you're saying you're using an in-house lab, they have said, well, are they ISO 15189 certified? And they say, no, they're not certified, but they do comply with the relevant regulations. And then if you do use one of these in-house institutes, so similarly to FDA's AI output, where they asked us about regulatory status, the EU authorities will do the same. So you have to provide the Health Institute information to the authorities during the submission of the clinical trial application. So these performance evaluation applications, I mentioned, think of it like an IDE. They are the only way that you can test interventionally in a US lab. There are reviewed and approved by the national competent authorities. The review timelines are not consistent. So for example, there's different review timelines if you go to France, if you go to Spain, if you go to Ireland. My favorite's Netherlands. They're usually faster. So there are additional considerations. When you go to FDA, you're dealing with one center. It's the same, right? Everything gets slowed down or everything's sort of running at the same pace, or there's a prioritization list. In Europe, it's completely different. They also have different expectations. So you'll get 3 pages from France's national competent authority, asking about software, asking about all these requirements versus, an Ireland competent authority may not ask for anything. So there's a lot of differences, and they need to work this out. There's actually a lot of clinical trials moving away from Europe because this has become so burdensome to get these through. With the RFIs, which honestly really increase with inexperience, it can take up to 9 to 12 months to get these through the authorities. There's a lot of documents. So just like the investigational device exemption, these are all the acronyms that you'll have on this slide with all of the required documents that you have to submit, just like an IDE. Of course, they have different words in Europe for analytical performance. So we mentioned, the importance of analytical validation terminology for FDA. They're similar, but you have to list all of these. And so something like TRUDIS, where there's a reference method, for example, and you're looking at the precision to the reference method through trueness, Europeans want that. So you use more of a checkbox where you make sure you give them all of these items because they're listed in the regulation. And they're very adamant about that if you don't include these. So it's not enough, for example, to say NA in Europe. You have to provide the rationale of why it's NA. Versus FDA, sometimes you don't have to do that as much. I always recommend it to give as much detail as possible, but I would say in Europe, it's very critical to pass these RFIs and be very clear. You'll even have the authorities ask you to reorganize parts of the submission just because they prefer things in a certain way. There's also a standard for these studies. So if you do find yourself doing an EU performance study application because you are doing testing in US for EU subjects, this is a good reference that you can purchase from Tech Standards, and it has a lot of details of what you should be doing for these studies. I bring this up, so I mentioned I was just doing EU and US. It did come up this week where someone forgot that UK wasn't part of EU. So there was Brexit and UK is its own country. So of course they have their own process and it's different than the EU and it's different than the US. So I just have this one slide as a reminder that don't assume it's the same process for UK if you're doing clinical trials that it is for EU. They have a distinct process. So now that you've got all your trials up and running, hopefully, no RFIs, everything's running smoothly, you may want to think about commercial in vitro diagnostics. So what about taking these to the finish line? So in the US, we have class 1, class 2, class 3. Those are our designations. And class 2 and class 3 are probably what functional precision medicine tests will fall under, moderate to high risk and high risk. Companion diagnostics can be either class 2 or class 3. Tumor profiling assays, for example, are often class 2. So it's important to understand if you are taking this to the finish line with the FDA, are you class 2 or are you class 3? And then companion diagnostics. So your functional precision medicine test could be a companion diagnostic. And there are considerations. For example, if it's being used to identify patients who are most likely to benefit from a specific therapy, or to identify patients who might be at increased risk for serious adverse events, that it could fall under a CDX. And we have distinct regulations, which I'll go over very quickly. This is the example labeling for therapy. So on the left, you have like a Keytruda label where you see this terminology as determined by an FDA approved test. On the right, you have a non-oncology example. And on the drug label, we have an agnostic label where it says using an FDA approved test. But differently on the CDX label, so this is the example from Foundation Medicine, you have to list the drugs. So you can even see the disconnect where a drug label is open and agnostic to using FDA approved tests versus the diagnostic labels will call out the tests. So there's labeling considerations as you are developing your test for certain therapies. It's always the same stuff that they're looking at, FDA analytical validation, clinical validation for IVDs. So you can do the precept that we mentioned earlier, not only for an investigational device exemption discussion, but also if there's additional data that's required to market these devices. This is just an information slide showing you cost of taking these through. So I mentioned earlier, one of the big issues for LDTs was how are we going to afford to take all these tests through? So you can see the MDUFA fees, for example, for a class 3 are $540,000. So that's just paying that to take a test through class 3. It's cheaper for class 2s, as you can see here. There are some differences. Class 2s have less regulatory burden. 510K is a pathway that we use when there's a predicate. So there's something out there that we can compare ourselves to and we demonstrate substantial equivalence versus de novo is more of a moderate risk where you may have to generate clinical data. And this would be the discussion you would have with FDA of what level of evidence would be needed for the test. Again, this isn't in the clinical trial setting. This is if you were to take it as an IVD through FDA. There are templates now, so it's very nice because you know what's expected. So if you are thinking about bringing your product through FDA as a class 2, class 3, I recommend looking at these templates, thinking with the end in mind, where you see everything that's required from FDA, and you can submit it through their portal. CDRH was pretty slow at becoming electronic, and so we've just, these submissions are still voluntary for PMAs, but I highly recommend using the templates because at least you know what needs to be in there. They can break sometimes, especially with interactive reviews. So sometimes you want to work offline in all your documents and pull it into the templates. There is also a big announcement. So you may have heard yesterday, Dr. Richard Pazder was now, he's now the CDER director. He wasn't too fond of CDXs, individual CDXs for every single therapy. He thought it was pretty cumbersome. So there was a CDRH reclassification announcement in January 2024 that was made, and we were all excited because we thought companion diagnostics would no longer be burdensome class 3s and would become moderate. But nothing really happened since then. But given the new FDA, I'm hoping that we see some reclassification because this would really reduce the burden for all of your tests as well as companion diagnostics out there. So just to close it out, in the EU, of course, it can't be class 1, class 2, class 3. That would be too easy. They have 4 buckets and they have letters: class A, class B, class C, class D. Most companion diagnostics and probably functional precision medicine tests are going to fall into class C, where they go through a notified body. And they do consider them to have a high individual risk and moderate public risk. So they want to see the technical documentation. They want to see the quality documentation. So that's what you can probably plan for Europe if you do take these. And there is one example. So this test is a functional precision medicine test that has gone through CE IVD marking. So I include it as an example. They did do some clinical trials, and this is available on the EU market. So it might be a good case study, to look at if you are thinking about the EU market. So that was a lot. So just to summarize, I really think it's important to ensure proper planning to avoid clinical holds. If if FDA has a lot of RFIs, if you have an investigational device exemption, it can slow you down. And I've seen it slow down companies and clients up to 2 years sometimes trying to make it, through this mess with FDA. So, hopefully today there's some, ideas for how you can potentially engage with FDA, interact with them. If you are going to be submitting INDs, if you think you might get an IDE, you can take opportunities to educate FDA. They're not experts with your tests, so it's good to kind of educate, go to them, have pre-sub meetings. You can have even information meetings with them that don't have to be, totally focused on questions. I didn't cover that today, but that's something you can look into in some of those guidances. Don't forget about the other laboratory regulations. A lot of clients come to me and they're like, oh, we didn't know New York State has extra regulations. And so then it can become an issue really fast. So just think about all those regulations for your studies that you're doing. Also using correct terminology. That's the biggest issue I see with submissions, is just not using the terminology that the regulators expect. Always consider both drug and device because they're not experts in both. So there is going to be a little bit of education. And then if you get through all those clinical trials and you are thinking about commercial in vitro diagnostics and taking it to the finish line, think about planning for the right regulatory class, monitor the regulations because there can be changes which are of benefit, right? If there's a least burdensome process with these reclassification announcements. See if you do have a companion diagnostic. There's different requirements for CDX. There's usually more interaction for CDX, and use the FDA templates as much as you can just to streamline the process. And if you have any questions now, feel free to ask them, and you can always email me too. Like, I'm happy. I really want these types of products to get to patients, and we shouldn't let regulations are important, obviously, but we don't want like not understanding the regulations to hold up, right, our development. That's not great.
Thanks so much, Kelly. That was great. Just jumping to the questions. So we had a couple in the chat. Mike, I don't know if Mike, you wanted to read your question out or you want me to go for it?
Oh, I can go ahead and ask. It's probably better. Mike Perricone and Kelly, we actually had a long discussion with you a couple of years ago.
Yeah.
And Kelly had the walls written. Just she, I still have pictures of our whiteboard with you. And it was a great talk and really important. I actually have, it's probably not the best question to start with, but I have a question not on the regulatory, but on the adopter side. So how are the pharmaceuticals, in your view, adopting these tests within their clinical trial? And what are the hurdles for them when they're considering including like a functional precision medicine test?
I would say in the past 2 years, it's harder to convince them with their budgets. I would say a lot of translational budgets in pharma have been reduced. And so I'm seeing them sort of do very minimal biomarker efforts, patient selection efforts if possible. So I'm not seeing, I would think, as much adoption as I would have seen about 2 years ago. I'm not sure if that's your experience from working with pharma. I just think less money. I do think it depends on the indication as well, right? Of these, I would say right in the heme space, they're probably a little bit more open to it than potentially the solid tumor space. But I'm not seeing, I do think the budgets and just the competition of pharma is affecting things a bit
Hmm.
for all my
Okay.
translational projects.
Oh, thanks for that input.
Yeah, and we had a question from Rebecca Wegman. And she had a question about RUO and CE-IVD tests. Can you use those in it if you're doing an ex vivo drug response assay?
Absolutely. So you can always use research use only reagents in CLIA tests, in in-house European tests, and CE mark tests. The trick though is, so you have to qualify them. So when you use a research use only reagent, it comes under your quality system. And so for example, do you have certain specifications when you order that reagent? That's important, and have all that documentation on file. So it is the one issue with RUO that I was sort of bringing up earlier is the problem is FDA can go after RUO manufacturers. So they're not going after the laboratory per se, but they could say to a manufacturer. So the warning letter example was a ELISA kit. I forget what the biomarker was, but they were saying, oh, on your website, it's suggesting it's for clinical use. So if an RUO manufacturer doesn't really clearly state this is for research use, The FDA, for example, can go after them, and the EU authority as well, obviously, and take that off the shelf. So then you're left with a CLIA or a CE-IVD test that has no reagent. So I always say use the reagents you want, qualify them, make sure they're high quality reagents, but have backup plans. So if you are using your favorite antibody, just make sure you have a backup in case something happens to the supply chain. But you are able to use it. It's just you're responsible for qualifying it for use in your in-house or your CIVD test. And the notified bodies, so when you do the CIVD and take it through the notified bodies, they do ask a lot of question about RUOs. So for example, mesoscale discovery, which is used for a lot of immunoassays, so many questions on that RUO. So be prepared to answer more questions if you're going to the authority, like a notified body with RUO, because they know you don't have control of it. But you can say, I mean, a lot of assays are single site IVDs where they are controlled in a laboratory environment. So you can use that to your advantage. You can say, we do lot qualification. We have these specifications. We test, prior samples. So you can justify it. There's just more paperwork for it versus using like a class 1 reagent, a class A reagent, or an ASR. Those are easier because they have more regulatory, documentation with them.
Yeah. Pam Becker, did you have a quick question?
Oh, no, I just wanted to say thank you so much for, this was a fabulous, it's fabulous to hear your presentation, and you've mastered so much of the details that are so important for us trying to do the work. Thank you so much. Really
Thank
appreciate it.
you. And thank you for your tests. Hopefully we have more of them getting to clinical trials.
We got like 1/2 minute left. Anybody else have another question they want to pop in there? All right. Well, if not, then thanks again, Kelly. That was really informative and really excellent to have your advice on this area. Thanks so much.
Perfect. Thank you all so much.
Oh, and don't forget to,
Thank you.
I think one last thing, if you're going to vote, you do have to join. You have to be a member to vote. So just go ahead and register. But for associate members who are students, things like that, it's free. So I think I was supposed to cover that right, Aaron. Thanks so much, everybody.
Thank you.
Goodbye.
Bye.
All right, bye-bye.